Hello there and welcome to this Biology Mind A-Level tutorial on cell division, where we'll be having a look specifically at chromosomes. So our key learning objectives for today are number one, to have a recap of the definitions of chromosomes, genes and DNA. And number two, to have a look at the structure of chromosomes. So let's get cracking. So a chromosome consists of lots of DNA molecules. It's one long strand of DNA molecules. Now, a DNA molecule has lots of genes, and a gene is made up of base pairs. So from largest to smallest, it goes chromosomes, which is a lot of DNA, and DNA is composed of a lot of genes. And genes are composed of a lot of bases. So you can see that our DNA is really tightly wound up into this chromosomal structure, and a section of DNA is known as a gene. So in humans, DNA in cells is wrapped around proteins called histones, which results in a structure called chromatin. And this chromatin is then tightly wound to form the chromosome. Now, humans have 23 different chromosomes which exist in pairs, which means that they have 23 pairs from chromosome number 1, chromosome number 2, down to chromosome number 23. Now, we are diploid organisms, which means that we have two sets of each chromosome. Hence, there are 46 chromosomes, because 23 times 2 equals 46. So there are 46 chromosomes overall, or 23 pairs, if you want to talk about them in terms of their pair numbers. Now, these are the different terminologies used. So I said that humans are diploid organisms, so we have 2 of each chromosome. Organisms with only 1 of each chromosome are haploid. Organisms with only 3 chromosomes are triploid, and organisms with only 4 chromosomes are tetraploid, where tetra means 4, tri means 3, di means 2, and hap is for 1. Now, each copy of a particular chromosome is called a chromatin. It's called a chromatid. Slip of the tongue. And so, let's say this is our chromosome. So you can see that each copy of the chromosome, because remember this is 2 copies here, is called a chromatid. So this is one chromatid and this is a second chromatid. Now, in humans, each chromosome is made up of 2 copies. So a chromatid has 2 arms which are held together by this central point called a centromere. And in most eukaryotic cells, 2 sister chromatids are joined together at this centromere. Then these orange regions, which are just these bits at the ends of the chromosome, are called telomeres, and we'll talk about them in a few slides' time. So the key words for you to remember here is that this is a chromosome, this is a chromatid, these are sister chromatids, and this is the centromere. Okay, so during the S phase of the cell cycle, the cell replicates the DNA, and DNA replication results in 2 copies of each chromosome. So since we normally have 46 after S phase, the cell will now have 46 times 2, which is 92 chromosomes, making it tetraploid. And the new copies of chromosomes are called homologous chromosomes. So the copied chromosome pairs with the one that it was copied from, and those are homologous chromosomes or homologous pairs. And ultimately, the chromosomes will be evenly split during mitosis, with each daughter cell splitting that tetraploid up. And overall, each daughter cell will have 46 chromosomes. Now let's come back to those bits on the end of the chromosomes, or chromatids that we called telomeres. So they have a very specific role in chromosomes. They protect the chromosomes during cell division. So telomeres are just disposable sequence of repeated nucleotides at each end of a chromosome. They don't necessarily code for any proteins. Now, the reason why I say telomeres protect the chromosomes during cell division is because in cell division, each successive chromosome replication cuts off a tiny bit of the chromosome at the end. So the chromosome gradually becomes shorter and shorter. Telomeres mean that they get shortened rather than the actual coding part of the chromosome. So the genes in the chromosome remain protected, because remember, this is where the genes are from here to here, and this is the telomere region. So if we're shortening the chromosome from the ends, we're going to slowly eat away into that telomere region, and we're going to keep those genes in our chromosomes protected. So as we age, that telomeric region is going to become shorter and shorter. Okay, so that's our tutorial complete for today. We've covered the structure of chromosomes, so I hope you will really understand that before we then move on to our next few tutorials, where we're going to be talking about the cell cycle and mitosis. Well done for following through today, and I'll see you for the next tutorial.